Yeah, I was looking at, I was trying to find that too.
What blood vessels do we find only in the cortex? Interlobular, also known as the cortical radiate. Okay. What else? What branches off of these vessels? Afferent arterial. And then that becomes glomerular, or the glomerular capillaries. And what does that become? Efferent arterial. And in the cortex, what does that become?
Oh,
Yeah, very, very what? Very tubular. Or trapid tube. Okay. What blood vessels are found only in the medulla? Interlobar. We're going to call arcuate vessels in between because they're right at the opposition between the cortex and the medulla. Okay. What else? We find only in the medulla.
the vas rectum.
Vas rectum. Okay. So then what structures would we find in the cortex? Optical convoluted tubule. All right. Depending on the straight, if we talk about the straight distal as that part that's right, just proximal to the macula densa between the afferent and efferent, yeah, but not the straight ascending. Okay. So straight and convoluted. Distal. we find only in the medulla that's the what? The nephron loop? And a cortical nephron that can also be, so the nephron loop can be in both, okay? So ascending and descending components of the nephron loop can be in both. What else can be in both? Well, we're gonna just leave that as in between the two.
Oh, the collecting duct?
Collecting duct could be in both. So thinking about the collecting duct, what would only be in the medulla? What part of the collecting duct is found only in the medulla?
The proximal tubule?
No, that's not part of the proximal convoluted tubule, it's found only in the cortex, but the papillary duct.
Papillary duct.
So the collecting duct starts in the cortex, passes through the cortex into the medulla, but at the very tip where several collecting ducts can come together, we would find the I need a different color, but oh well, this is what I have. We'll find the papillary duct will be found here as well. Okay. So don't memorize this list, but as you diagram the structures or mentally move through them, all right, recognize where we're finding it. 'Cause that will help you with the histology structure too, as far as recognizing where you are, what structures you might find there. Okay? All right, question number 2 then. Which of these structures can be found in both the cortex and the medulla? How many correct answers did you have here? Two. Good.
What's that, under cortex, it's straight, it's that second one on the picture, that she wrote straight, something distal.
Which one? The red one or the green one?
The green one, under cortex, it's straight, something distal,
Oh, straight slash convoluted.
convoluted.
All right, so we're gonna start out in the process of urine formation by looking at the first event, which is the filtration, okay? And we reviewed the definition of filtration by saying it was the separation of particles by size. So we have to set up what that, filtration sleeve or sieve is going to be formed of. So the 3 main events that occur in urine formation, modifying of the plasma of the blood, is filtration, reabsorption, and secretion, all right? So filtration is the movement of those particles small enough to leave the blood to go into the capsule. And at that point, we call it filtrate, not urine, okay? And as it moves through the rest of the nephron, primarily in the proximal convoluted tubule, and in that region based solely on concentration, not using hormones or drugs or anything to change it, we move material from the plasma back to the blood. And this would be our peritubular capillaries that are going around and all over the depth of the proximal and distal convoluted tubule, all right? So this is indiscriminate except for size. Whether the body needs to get rid of it or not, it's gonna get filtered through. And then we start the process of returning everything back to the blood except for what's going to be excreted primarily, and that's the reabsorption, all right? Then, we move some additional material into the filtrate. So for instance, if the body plasma is too acidic, we're gonna move hydrogen ions into the filtrate. We'll talk about that next week. if the body needs to get rid of additional components, say you've been taking antibiotics or drugs that are being broken down in the liver, those byproducts, additional byproducts will be added to the filtrate. So that's called secretion, all right? So reabsorption is moving from filtrate back to blood, and secretion is adding additional material that would have already been filtered, but adding even more to the filtrate, all right? So we're gonna look at the process of filtration first and look at the anatomical components, and then we'll look at sort of the mechanics. So make sure you have a picture of what's on the board if you haven't written it down yet, because I'm gonna erase it.
Can I start a
Draw it, yeah.
On the nephron, or not the nephron, the glomerulus that you brought up?
Uh-huh.
So there's the visceral of, on the
Capsule?
capillaries, and then on the outside of the,
Uh-huh.
inside of the capsule is the parietal.
So this would be the parietal layer. So your question is?
I was just wondering if it was like parietal something, or just parietal
It's
layer?
just the parietal layer of the capsule.
Okay.
Yeah, and we don't have a special name for it like we do the peritoneum or the pericardium or anything like that, okay? So this structure that you see here is the one that I showed you on Tuesday night based on this model, all right? So this would be the distal straight tubule of the nephron, that we're gonna be talking about how it leads to the change in the diameters of these vessels. Here would be the parietal layer, and where the vessels enter into the capsule is called the, do you guys remember? It looks like the hilum, but we had two poles. This was the vascular pole and the urinary pole, although it should be the filtrate pole, because it's not really urinary, okay? So, we're gonna talk about this structure here, which is called the juxtaglomerular apparatus, and the regulation of this pressure. But, as we look at the structures, we have the simple squamous epithelium of the capillary, and that's covered by the simple squamous epithelium of the visceral layer, specialized cells that we call podocytes. Anybody know what poto refers to? We usually see it spelled P-E-D instead of P-O-D. So pedometer, pedestal of the vertebra, pedestrian. What would that refer to? Foot. Okay, so these are specialized simple squamous epithelial cells that have feet processes that rest on the epithelia. So may I erase that board now? And we'll diagram some of these components. I don't know how this board is so clean after a whole day of teaching in here. So here's what the glomerular capillary, we saw this a bit on Tuesday. So here would be the simple squamous epithelia of the parietal layer. And then here you can see some of the red blood cells, but a whole bunch of other cells in between, all right? So here's what it would look like, which is the capillaries. So those are the glomerular capillaries without the epithelial cells of the capsule. And a couple of places like here, you can see the large afferent arteriole and the smaller efferent arteriole coming out. But for our filtration membrane, we need the additional visceral layer of the cells of the capsule, and these are the podocytes. So they're not our typical simple squamous epithelia that we saw in the respiratory membrane. We have a large area where we have the nucleus, and then the podocyte has branches that spread out, and then off of those branches are the foot processes that come down and rest on the basement membrane. So we have two sets of basement membranes because we have two sets of epithelia. So I'm going to draw this with the capillary down here. And the capillary has its own nucleus. And we'll draw the other capillary down here, although I'm not going to be the other side of the capillary, all right? And then there are gaps within the capillary. So this is all one capillary cell, all one simple squamous epithelium. This would be the core, or the fenestra. Remember we talked about fenestrated epithelia for the capillary? So this structure is known by the fancy word fenestra. A-E makes it plural, which just means pore, all right? Now, as you recall, the epithelia has on its basal surface basement membrane. All epithelia, that's one of our characteristics of all epithelia, is basement membrane. And in the basement membrane, so if we have our typical epithelia cell here, we have a layer of the basement membrane made by proteins of the epithelium, and we have a layer of basement membrane made by proteins of the connective tissue, the fibroblasts. And so we call this the basal lamina. And this the reticular lamina. Recall that from Bio 430? At least there is a basement membrane there. All right, so we're going to have the basement membrane, and it's continuous. From the epithelial cells, all right? Now this would be the, I drew it backwards. Let me change the diagram here so it fits what I drew on the. So this would be our epithelial, capillary epithelial cell. And we would see the same thing down here. I'm just not going to draw further components. Then we have the epithelial cell of the visceral layer, and that's what you see on the screen right now in the scanning electron micrograph. That's also a simple squamous epithelium. And it has these podocytes. Well, pedocytes. And then the whole cell is known as the podocyte. And then up here, we would have the parietal layer of the capsule. Podocyte is part of the visceral layer. Okay. And they interdigitate. So here would be another podocyte. I'll put in the hash marks just to clarify which one is which. All right, so in between the foot processes, let me put up the basement membrane of that epithelium. So in between the foot processes, we have a filtration slit membrane. So it's not a completely open space, but it's made up of some proteins that have negative charges. And those negative charges affect the movement of particles through here. So we have the fenestra, we have the basement membrane, and we have the filtration slits. Where's a good place to diagram that? So let me draw a filtration slit up here for clarification, next to the clarification of this basement membrane. So this would be podocyte A, podocyte B, podocyte A, all right? And those proteins create a structure known as a slit diaphragm. So this is our filtration membrane. Remember we had a sandwich of epithelial cells that we called a respiratory membrane, and we're looking at the alveoli. And we had our bread, which the two slices of bread were the simple squamous epithelia, and the filling was the connective tissue. And that determined the distance of diffusion from the alveolus to the capillary. So here we have a filtration membrane. Two slices of bread are the simple squamous cells of the capillary and the podocyte of the capsule, and the filling is our basement membrane, the two basement membranes for each different epithelia, all right? And then this space here would be the capsular space. And it contains filtrate.
What's the difference between the filtration slit and the slit diaphragm?
The filtration slit is the gap, and the slit diaphragm are the proteins that kind of create a membrane across that gap. By membrane, I don't want to overuse the word membrane. A thin layer that material has to get through. It's not just an open space. The fenestra is an open space, but the slit diaphragm is a barrier through the filtration slit. So the size,
So the whole thing is
the filtration membrane.
the whole
So
thing is
these three layers, the epithelial capillary, the epithelia for the podocyte, and the basement membrane between, those create the sandwich that is the filtration membrane. So like the respiratory membrane in the lung is two simple squamous epithelial with connective tissue in between. This is two simple squamous epithelial with basement membrane in between, and that's the filtration membrane. That's what, say, a molecule of amino acid has to cross to get from the plasma into the filtrate. It has to go through that pore, through two sets of basement membranes, through the slit diaphragm of the filtration slit, and then finally it's in the capsular space. So here's looking at it from within, like a cross section. This would be a longitudinal section through the capillary. Here's a cross section. So here's the fenestra or pore. Here's the basement membrane of the two different epithelia. Here's the pedicels of the podocyte cell, and that darker blue would be that filtration, the slit diaphragm. Okay. So the basement membrane and the slit diaphragm affect what passes through by charge. They have a negative charge, so they tend to repel components that are more positive, and the pores are the size determinant. Yes, Caitlin?
When you say the three layers of the filtration membrane, do you say filtration slit as one of them, or filtration?
The layers are the epithelia and the basement membrane in between.
Yeah.
The filtration slit is just part of the podocyte layer. Just like the fenestral, or the pore is part of the capillary layer. So here's our filtration membrane. Capillary cell of the epithelial cell of the capillary, fused basement membranes, podocyte. Okay? And then we break those down into smaller parts, like the pores and so on. Here's another more molecular view. So here would be the capillary epithelium with the pore. Here would be the capillary basal lamina, capillary reticular lamina, fused with podocyte reticular lamina, podocyte basal lamina, podocytes, the pedicels of the podocytes, and the filtration slit with the slit diaphragm as the very between. Okay. The words nephrin and podocin, you don't have to worry about the podoclexin. The nephrin and the podocin are negatively charged proteins that form that slit diaphragm. They're part of this circle here. So we would have plasma here as our fluid, where I wrote plasma there. And then up here we would have the filtrate. Okay? So now that we've looked at that structurally, let's take a look at what it looks like in an electron micrograph. Okay? So here's what we saw in the first picture while I was diagramming it on the board. And here it is in a thin section. And notice all the proteins that you see here, all these flocculent materials. So this is the plasma. Here is the epithelial cell of the plasma. And you can see a light, dark, and light area. I think I have a larger view. So here would be the plasma would be here. Capillary cell of the plasma, you can see the fenestra or pores. All right, here's the light area of the basement membrane, which would be the basal lamina made by the epithelial cell. The dark region is the reticular lamina of both layers, both epithelial cells. Here's the basal lamina of the podocyte, and then you can just barely maybe see a dark line right there, right there, right there. So this would be the filtration slit, and that dark line represents the slit diaphragm. So plasma would be below here, filtrate would be up where the letter V is. Okay. So essentially, back to this diagram, we're looking at it from this view. We're seeing the capillary pores, basement membrane, podocytes with the slits. All right, so for this question, I'm asking you to identify the light region where that black arrow is. So essentially this whole light area right here. In the first picture while I was diagramming it on. So what the force is doing, rather than gravitational force, what's the force that's pushing the small particles through this membrane? You're still going to filtrate if you're standing on your head. So the force comes from the heart. So it's blood pressure, hydrostatic pressure of blood pushing against the capillary wall is going to push small particles through the pores, through the basal lamina, through the fused reticular lamina, through the basal lamina again, and then through the filtration slits. So there are actually some diseases that occur when these proteins, the cell doesn't produce some of these proteins and that filtration slit is damaged. There are certain kidney diseases that occur as a result when components of the plasma get through that are not supposed to get through into the nephron. All right, so may I erase the diagram, the filtration membrane on the board? Okay, and we're going to look at those pressures. We have both positive pushing pressures and pressures that are pushing back. Okay.
I think it was in the laminas.
So this is showing a simplistic arrangement of the capillaries. It's not showing the intimate relationship between the podocytes and the capillaries, but a more simplistic arrangement of that glomerulus and the capillary component. So wide vascular fold here, and we have the capillary coming in. So in the capillary, we have blood pressure. All right, it's not very high, but it's higher than other capillary blood pressure on the body. So the length of the arrow is representative of the degree of force. So we call this the glomerular capillary pressure. Okay, and And that's going to be affected by systemic pressure. So the higher the systemic pressure, the higher the glomerular capillary pressure, unless we change the diameter of the afferent and efferent arteries. So this is going to be glomerular hydrostatic pressure, which is the same blood pressure. All right, and that's typically about 50 millimeters of mercury. So in relationship, systolic pressure on average is around 120 to 70. And so we're saying this is 50, all right? So we'll put that formula over here. Glomerular blood pressure is 50 millimeters of mercury. All right, so as that pushes fluid into the capsule, we start to build up the fluid pressure, the hydrostatic pressure of the capsule, all right? Just like as you push flow air into the balloon, the more gas, the more air you put into the balloon, the more pressure is pushing on the balloon walls, and it starts to expand. So the more air you put into the balloon, the more resistance there is to putting more air into it, all right? Because you're having to pack that, increase the pressure, because you're increasing the number of molecules of gas already that you're adding to the balloon. So the capsular pressure is going to be about 10 millimeters of mercury. So that's pushing back with the fluid, all right? And we're pushing back. And so this is a negative because it's pushing back. And this is capsular hydrostatic pressure, or blood pressure. It's not blood, but hydrostatic pressure. It's the fluid of the filtrate. Now, what would happen to that capsular hydrostatic pressure if there is some kind of problem with the filtrate flow or urine flow leaving the capillary ducts? Let's say that there's a large stone in one of the minor calyces. What's that? A large kidney stone, one of the minor calyces. What's that going to do to capsular hydrostatic pressure? Going to increase it or decrease it? Going to increase it because it's not allowing the filtrate to continue flowing through, so it's going to start backing up towards our filtration membrane, all right? So that would increase it. All right? Anybody know what colloid pressure is? Maybe if we change it, use just the osmotic, what would that be? I'll give you a hint. Albumin? Okay, so what does albumin do? When we were talking about the lymphatic system, and I introduced the lymphatic system to you, we talked about the osmotic pressure of the capillary. Did that increase the amount of fluid leaving the capillaries or decrease the amount of fluid? It decreased it because it has a pulling effect, all right? So the more albumin we had in the blood, the less fluid was lost to the extracellular fluid. And so we talked about how the number of albumin molecules increased as we moved to the venous end because of the loss of fluid, that loss of fluid but no loss of albumin kind of increased the concentration. So if we have proteins, we have albumin present in the capillary. That's going to have a force in this direction. And that's going to be a counterbalance to the hydrostatic pressure. So 2 is our capsular hydrostatic pressure. And 3 is our osmotic pressure due to albumin, capillary osmotic pressure. Okay? So that's typically minus 30. when we're done, the net filtration pressure, okay? That net filtration pressure ends up being approximately 10 millimeters of mercury. So we started out with 50 as the force pushing the small particles through the filtration membrane, resisted by the force of fluid already in the capsule, resisted by the osmotic pressure of the albumin proteins in the blood. So we come up with a net filtration pressure of 10 millimeters of mercury, okay? Now, if because of increase in blood volume or the cause maybe not known of increase in systemic pressure, if this number is higher, this number is higher, okay? If there's liver disease and there's a decrease or lack of protein in the diet and there's a decrease of albumin, remember that resulted in increased edema, then this number drops and this number would go up, okay? So those are the three major factors determining the net filtration, the net force that is still in a positive direction, still in a positive 10 millimeters of mercury moving fluid from the capillary into the cap, from plasma to filtrate. So that's what we start out with, okay? The assumption that we start out with. So now we have mechanisms that we can modify this. All right, we talked about the diameter of the afferent and the efferent arterioles. And when we talked about blood pressure back with our cardiovascular system, we talked about the hormones, aldosterone, and atrial natriuretic peptide, and antidiuretic hormones. We're going to mention those again as far as how we release them, and then on Tuesday we'll talk about more of the role of ADH in the collecting that. All right, so which of the following oppose the movement of fluid from the capillary into the glomerular capsule? You can read that in the math problem I put up on the board. Okay. So what reduces the glomerular filtration pressure, the hydrostatic pressure, from 50 to 10? What would capsular osmotic pressure do? I have capsular osmotic pressure up on the board anywhere? No. To have capsular osmotic pressure, we would have to have a damaged basement membrane. Because capsular osmotic pressure is due to protein. Proteins are too large to get through our filtration membrane. So if we have a damaged filtration membrane, then we can have capsular osmotic pressure, which is going to pull more fluid through. Okay? Because normally proteins are too large to get through the fenestra and through the filtration system. So how many answers are correct? Two? Two. Anybody not understand this question? Where it's coming from? All right, so the main thing, this makes pretty much sense, but be able to think of factors like the kidney stones, or change in the albumin, or damaged filtration membrane that might modify these relationships. All right, so here's a more detailed look, a little more similar to the model that we had. So we're still at the vascular pole. All right, we have the afferent arteriole, we have the efferent arteriole, smaller diameter. We have our glomerular capillaries, and the yellow structures around here are the pedicels of our podocytes. We have our distal straight tubule, and in the region, remember I said it has to come through between the afferent and the efferent arterioles, and some artists will draw it, and so coming back up through, it will be way over on the opposite side. But it has to come between the afferent and the efferent to create this juxtaglomerular apparatus, which is sensitive to the requirements for changing and modifying pressure. At the area of the distal straight tubule facing the glomerulus, the cells become columnar instead of cuboidal, and very narrow, and that brings their nuclei close together. So that is called the macula densa, right, which means dense spot. And on the model, it's seen as this area right here, okay? Facing towards the vascular pole, facing the capsule. Okay. Now, in addition to the typical three components, the macula densa, in this afferent arteriole are specialized cells known as granular cells or juxtaglomerular cells. And then we have extraglomerular mesangial cells in here. So let me erase this portion here, and we'll redraw that on the board. So this is the afferent arteriole. And in the smooth muscle of the afferent arteriole, we have cells that have granules of an enzyme known as renin. Okay? So this component of the juxtaglomerular apparatus are known as granular cells. The older name, and one that you still might see, are juxtaglomerular cells, but that makes it sound like they're the only cells of the apparatus, and they aren't. So these are smooth muscle cells that contain this renin that is going to be, when it's released, starts changing blood pressure relationships, all right? We have the efferent arteriole leaving, which can change its diameter but doesn't contain any chemicals. Then we have our straight tubule, with the simple columnar cells. Known as the macula densa.
Does it make a difference that the picture on the board is the afferents going up? And the
So
efferents going
afferent is going into the capillary bed,
up? Okay.
efferent is leaving. E for exit.
But does it make a difference that that one shows the efferent
Oh,
going
no,
up?
it's just-
Oh, it can be either.
I'm
way.
sorry, I could have done it the other way around.
Okay, so it doesn't matter
Yeah.
which way it's going, you just have to know which way the fluid is
Yeah.
going. Okay.
It's gonna be flipped around. Sorry, I usually try to follow the diagrams. All right, then within here, we have our capillaries. Surrounded by the podocytes. And importance was only, they were recognized, but for a long time, nobody knew exactly what they did. And these cells are called intraglomerular mesangial cells.
Is that another word for mesenchyme?
Mesangial, no, well, it means tissue. Let me update what makes up our juxtaglomerular apparatus here. The granular cells, the macula densa, the extraglomerular mesangial cells.
So I think this is probably not gonna be on this test works, but
Sorry, she's saying extra, intra. There are extra, but nobody knows what they do yet. And those are found the intraglomerular are found here, okay? I'm not gonna label them because I'm not gonna test on them, but those are the, my words make sense. These are the extra, intra or inside, these are extraglomerular. Meaning they're outside. Glomerular mesangial cells. Okay. So, afferent arterial, efferent arterial, macula densa, intraglomerular mesangial cells, okay? Extraglomerular mesangial cells. We're gonna focus on them, the macula densa, the granular cells, and the intraglomerular mesangial cells. So the purpose of this is to affect that filtration pressure we talked about, all right? Whether or not we increase filtration force or decrease filtration force. I don't know if you can see, let me turn the lights down. So see these red dots, pink dots? Those are the granules of renin, and why we call them granular cells, okay? Or the old name juxtaglomerular cells. So it's the smooth muscle of the afferent arterial. Laces cells is just another name for those extraglomerular cells that nobody knows exactly what they do. A better close-up view of the renin, granules of renin, and the smooth muscle of the afferent arterial cells. All right, so we're going to describe 3 different events that can occur to change the flow and filtration pressure. So, remember how we said our Frank Starling law? And we said the more we stretch the ventricular muscle, the greater the force with which it contracted. All right? So, when the afferent arterioles are stretched, and that would be due to a greater blood volume flow, meaning higher blood pressure, right? So, if there's a greater increase in cardiac output and more blood flows towards the kidneys, to protect the kidneys, the afferent arterioles will constrict with that stretch. So that might help you if you think about the Frank Starling law. So when we stretch the ventricles, they contracted harder for a different reason, okay? Now the afferent arterioles, when they get stretched out by a greater blood volume, will constrict. That functions to protect this filtration membrane of the capillary cells and the photocytes, all right? In contrast, if blood pressure is very low, not enough blood volume in the body, and afferent arterioles collapse because of the lower blood pressure, then they will dilate to increase the flow. It seems counterintuitive. You think they would constrict to increase pressure, but that decreases flow. We want more blood flowing into the capillaries. So if blood pressure drops, these vessels dilate to increase the amount of flow going through.
And using the ventricles to bring them?
No, I'm just saying, I was just relating
Oh,
to
okay.
that effect, okay? So, if we have a lot of force, pushing on the walls of the blood vessels, they will constrict to decrease the amount of flow through the capillaries, protecting the capillary filtration membrane against damage from high pressure. If blood pressure within the afferent arteriole is low, they dilate to encourage more flow into the capillaries. Okay? So constriction of afferent arteriole decreases glomerular flow. Dilation of afferent arteriole increases glomerular flow. Okay? Like if I have a small tube, I can't put very much through it. If I have a larger tube, more fluid can flow through that. All right, so that's what's happening in A. Now, that's just due to the effect of the stretch on the cells. In a similar response, in a similar effect, but from a different response, if pressure is low, because remember the filtration doesn't occur very well with low pressure. We need that force pushing small particles through the filtration membrane. So if the pressure is low, in addition to that dilation that occurs, all right, the glomerular cells will release renin based on detection by neuroreceptors of low pressure. So the effect is the same, but from a different response. So the renin, we'll talk about what renin does in just a moment, but the renin is going to work to increase the systemic blood pressure. Okay? Downstream, we're going to talk about that. So renin right away is not itself going to change the contraction or dilation of the vessels. It's going to affect systemic pressure, all right? Another way that the granular cells can release renin is through the sympathetic nervous system. So beta-1 receptors, all right, are what we had in the heart, and beta-1 receptors activated by the sympathetic nervous system will also cause the granular cells to release renin. And the third way is where we're going to bring in the macula densa. Okay? The macula densa can signal the afferent arterial cells to release renin by sending prostaglandins. This would work at a paracrine type of effect, a local hormone, prostaglandins, remember, come from arachidonic acid. But they're not having to get into the bloodstream, so they're just going to be released locally to a different cell type. So that was our definition of a paracrine relationship. Because the macula densa cells are not the same kind of cell as the smooth muscle cell. So let's see how we activate the macula densa, and then we'll go back over and review this. Okay. And you have this, I believe, in the written description in the lecture notes. So let's look at this picture first, and then we'll talk about. And I mentioned a tiny bit of it on Tuesday. Remember the flow of the filtrate from the capsule, from the capsular space. It flows into the proximal convoluted tubule. And 80% of our sodium is reabsorbed in the proximal convoluted tubule. You don't have to write that down now because we're going to talk about it in a bit. All of our glucose and all of our amino acids are going to be reabsorbed there, all right? And then that's going to continue to flow through the nephron loop in the distal tubule. And then it's going to come up the distal tubule into the straight tubule portion here and by these macula densa. You've got that sequence in mind. Now, as it's flowing through the proximal convoluted tubule, and sodium is moving from its high concentration to its low concentration into the cells of the proximal convoluted tubule. If the filtration pressure is low, it's going to move through that proximal convoluted tubule more slowly than if the filtration pressure is high. If the filtration pressure is high, we're filtering a lot of fluid. It's going to flow rapidly through the proximal convoluted tubule. That means it's going to be in contact with the microvilli of the proximal convoluted tubule for a shorter period of time. which means fewer sodium ions are gonna be reabsorbed. All right, so we're gonna have more sodium ions in the macula densa when it gets there. However, if the filtration rate is low, because our filtration pressure is low, then it's gonna flow more slowly. And the proximal convoluted tubules are gonna have more time to reabsorb more sodium. Thus, when the filtrate comes back up by these vessels through the straight tubule, the fluid, the filtrate's gonna have low amounts of sodium. So, low amounts of sodium is interpreted by the macula densa cells as meaning low filtration rate, more reabsorption of sodium. So in response to those low amounts of sodium ions in the straight tubule, the macula densa cells, and here's a histology view of the macula densa. I don't know if you can see the fine Renin granules there, but there's a close nuclei of the macula densa. Then they release the prostaglandins. Okay. And the prostaglandins go to the granular cells and cause them to release renin. Right? So we listed in 3 ways, and but that could 3 things, 3 mechanisms, or 3 methods that will trigger the granular cells to release renin. We haven't yet talked about what renin does. We'll get to that. So renin can be released. Go back to this diagram. Oh, it's going forward. Renin can be released when there is low pressure in the blood vessels. Renin can be released when the sympathetic nervous system triggers low pressure, triggers release. And these often occur together, okay? And then renin can be released because of the prostaglandins. So it can be released on their own just because of pressure changes. It can be released by activation of the sympathetic nervous system on those cells. And it can be released by prostaglandins moving into the cells. Would prostaglandins be soluble in the plasma membrane? It's made from a fatty acid. Arachidonic is a fatty acid. So like the steroid hormone, it's soluble in the plasma membrane. So it can go directly into the cell, all right, and it will trigger the renin. So without any of that, just because of dilation, we have that myogenic effect, all right? But then we have these chemical changes that can occur. All right, then we have the intraglomerular mesangial cells. And this is relatively new, understanding the effect by relatively like your lifespan, okay? The intraglomerular mesangial cells, so here's our capillary, here's our podocyte cells with the filtration slit membrane. Here's the intraglomerular mesangial cells. And it's kind of backwards, all right? What you think of. When the intraglomerular mesangial cells contract, because they're kind of a weird, they're not really a muscle cell, but they can contract. And when they do, they decrease the amount of basement membrane around the capillaries, and that decreases the surface, and I don't understand the physics of that, but it decreases the surface area, so there's less room for filtration.
Will you say that one more time?
Say that again?
Yes.
So contraction of the intraglomerular mesangial cells is going to decrease the surface area of the basement membrane, which decreases the filtration ability. So this is going to be the opposite effect of what we just talked about. So intraglomerular mesangial contraction is going to decrease filtrate formation. So afferent renin release is going to, this is all going to result in an increase in flow, filtrate flow. This is going to result in an increase in filtrate flow. Intraglomerular mesangial cells is going to result in a decrease in filtration flow. So that would occur if blood pressure is too high. Okay? Now, so the renin is in response to low blood pressure. The prostaglandins is in response to low blood pressure. Okay? glomerular filtration is in response to high blood pressure. We have one more component in response to high blood pressure. So again, if we have high filtration rate, we're going to have the prostaglandins are released due to low sodium. With a high filtration rate, the filtrate is flowing through the proximal convoluted tubule too fast to reabsorb much sodium. So when that happens, the macula densa cells release adenosine. That's an amino acid. And that functions to decrease flow rate due, down over again, due to high sodium, okay? So let me put this screen on blank so I can write on the screen board behind it.
Oh, so it's kind of like negative feedback type thing, like it prostaglandins.
So when blood pressure is low, all right, afferent arterioles dilate. That was that first effect that we saw that was called myogenic. That didn't have anything to do with the renin yet, okay? Then we had three mechanisms of renin release. It's going to result in an increase in systemic blood pressure, which we haven't talked about yet, how that works, okay, but just remind you. So the three mechanisms of renin release are baroreceptors. That just means pressure receptors.
But only when it's low pressure, right?
Pardon?
Only when it's low pressure?
Right.
Okay.
Mhm.
So when I'm like studying for the test, would I wanna study baroreceptors in the granular cell, or would I wanna
Baroreceptors
study
just
low
mean
blood
pressure detection. So the baroreceptors detect low blood pressure,
Okay.
and they cause the granular cells to release renin. Okay? Sympathetic neurons. Release norepinephrine, which binds to beta-1 receptors. On the granular cells, and that also triggers the release of renin. Okay, and then macula densa cells release prostaglandins after detecting low sodium in the filtrate. And the prostaglandins trigger the glomerular cells to release renin. Okay? So this is the only immediate effect because of low blood pressure. These are three mechanisms that are going to cause the systemic change through red and blues. Okay? All right? So,
But what about the other thing,
The
so
last
that's
thing you talked
released
about.
after the prostaglandins?
that's, so,
Yeah, it's underneath
At
it.
the-
yeah, that's what I'm gonna put on the, when blood pressure is high.
Okay.
So may I erase this side of the board?
Mhm.
And I'll put the when blood pressure is high on that side. Okay.
Do those cells do anything when the blood pressure is low?
The question was whether the intraglomerular mesangial cells have an effect when blood pressure is low.
So the reason for like high or low sodium in the filtrate is based off of the blood pressure.
Yeah, and that affects flow rate.
Okay.
So again, so when blood pressure is high, that glomerular hydrostatic pressure goes from 50, goes higher, and therefore there's a greater net filtration pressure, and it pushes more fluid and more particles into the capsular space. That increases the flow rate. It's like turning a faucet on with greater volume. There's more water coming out of the faucet, and that's going to flow faster through the nephron tube, through the proximal convoluted tube and the rest of the nephron, meaning there's less time for it to have contact with the microvilli, meaning fewer sodium ions are going to be reabsorbed. By the time it gets back through the distal straight tubule, there's more sodium in for the macula densa to detect. So the macula densa is responding to concentration of sodium ions. Concentration of sodium ions is low, it releases prostaglandin. All right, that causes more renin release. Concentration of sodium ions is high, it releases adenosine, which causes the change in the diameters, which decreases the amount of flow by constricting the afferent arteriole, dilating the ether. Okay.
So does adenosine
Yeah.
directly blocks the renin release or no?
Yeah.
Oh.
It just turns that system off and changes the muscle contraction. Okay? Ready for a quiz question on this? At first I had to cover up this side of the board to show the screen, but I don't want to do that till make sure you have that down. And I have this written on the PowerPoint, okay? And summarized in your lecture notes, but probably in a different sequence. So, imagine, think of something that, so the slower, you got water flowing through a creek, and it's got debris in it, all right? The slower that flows, the more time the debris has to settle to the bottom of the creek. And when it gets to a mile downstream, there's gonna be less debris in it. Yes?
So wouldn't that mean that low blood pressure would have higher?
So it means that low blood pressure means less fluid in the process of our filtration membrane, okay? I know it seems kind of backwards, but because there's lower flow, the flow rate is lower, think of again, the debris in the creek is gonna settle down, so when it gets to a mile downstream, there's gonna be less debris. The macula densa interprets that as meaning that the flow rate is too low, meaning there's not enough fluid, meaning the filtration pressure is low, meaning the volume is low. So it causes the renin to release, which after the quiz question we'll talk about how that works. All right. So macula densa interprets the low sodium. And low sodium is due to a slow percolating. Think of it going so slowly that more of the sodium is being removed from the fluid. A high flow rate means, so like that, you're on a raft and you're floating along a river and it's going really, really slow. You can step off that raft whenever you want to. If you're speeding down at 50 miles an hour, it's pretty dangerous to try to get off that raft and onto the bank of the river. So you're just going to stay on that raft and still be on it by the time you get to the mile downstream. So if blood pressure is high and it's pushing more fluid into the capsule and into the nephron, and it's flowing rapidly by all those sites where the sodium could leave, then it's still in the fluid by the time it gets to the macula densa. The macula densa detects that and goes, all right, we need to slow down the filtration rate, and it releases the adenosine. if the flow rate is so slow that sodium has a chance to be removed, then there's less sodium when it gets to the macula densa. And that's interpreted as we need to increase flow rate, and it releases the renin to increase blood pressure. Okay? So high sodium, high blood pressure. Low sodium, low blood pressure. And the macula densa changes that. If the blood pressure is too high, it wants to lower it. Blood pressure is too low, it wants to raise it, as far as the filtration force is concerned. Okay? Now, I was going to make both B and C wrong, but I decided that was too tricky. I've already played with your brain. So either B or C is, one of them is correct. We're talking about in the filtrate, passing by the macula densa cells. The answer's on the board, at least the interpretation. The answer itself is behind the screen.
Yeah.
What would you have to change to make A, B, and C correct? All right, so is it a component of the distal straight tubule? Yes. Does it respond to high levels of sodium by releasing prostaglandins? No. So how do we make that statement correct? Adenosine. Okay. And then C would be correct the way it is. Okay? All right, let's take a look at how renin works, and then we'll take a break. Pardon? Do we have a lot of We do. You guys ready to look at the diagram of renin? Okay. So a lot of little words, so I want to diagram this on the board. And not everything in here is new to you, because we talked about some of this, just not how we got to it, when we talked about cardiac output and blood pressure. So without the pictures, the liver makes a protein known as angiotensinogen, okay? Like it makes other clotting proteins. And remember when we were talking about clotting proteins, we went from fibrinogen to fibrin, and an enzyme caused that to occur. All right, so this is in the blood. It gets to kidney, afferent arterioles. Let me write that a little bit higher. And under those conditions that I talked about, currently behind our screen, if blood pressure is low in the afferent arterioles and our amount of flow filtration rate is low, then The kidney arterial granular cells release the enzyme renin. Renin is not a hormone, okay? If you read that it is, it's incorrect. Renin is an enzyme. And the effect of renin is to convert angiotensinogen to angiotensin 1. Okay? Now, angiotensin 1 can have some mild effects of vasoconstriction, but it's not as active as angiotensin 2. So angiotensin 1 then goes to the lungs, because all the blood flow goes through the lungs too, just like it goes through the kidneys, all right? So in the lung, in the capillaries of the lung, another enzyme is released, and this enzyme is angiotensin converting enzyme. And it further modifies angiotensin 1 to angiotensin 2. Okay. And angiotensin 2 then has basically 5 effects that cause blood pressure to go up. Okay? So angiotensin 2 causes norepinephrine, more norepinephrine to be released by sympathetic neurons. It's going to work on postganglionic sympathetic neurons, and they're going to make more norepinephrine. So blood vessels are going to constrict as a result of that, right? Remember, blood vessel dilation or constriction has to do with just the sympathetic nervous system, unless we're talking about the blood vessels, the arterioles involved in erection. So more sympathetic activity is going to result in increased vasoconstriction, systemic vasoconstriction, and that's going to increase systemic blood pressure. Do we want to increase the afferent arterial constriction in the kidneys though? No, we want those to dilate, right? Because we want more blood flow into the glomerulus. But if we increase vasoconstriction of blood vessels around the body, general blood pressure goes up, and that raises our capsular hydrostatic pressure, right? So number 2, it's going to increase water reabsorption in the kidneys. Just directly by affecting the water reabsorption. Okay? On its own, very similar to number 1, it increases vasoconstriction without the sympathetic nervous system. And then it increases 2 hormones that we've already talked about. It causes the adrenal glands to release more aldosterone. We'll talk about that effect on Tuesday, but aldosterone increases water reabsorption in the kidneys by increasing sodium reabsorption. And this is at the distal convoluted tubule, okay, so not the proximal. So this occurs after the sodium has gone by the macula densa. Because it's, you go, well, if that increases more sodium reabsorption, that would make the macula densa detect low sodium. But this occurs after it goes by the macula densa, all right? So this is post, the effect is post, after the macula densa. hormone. It increases the posterior pituitary's release of antidiuretic, and that effect is on the collecting duct. So 1 and 3 increases blood pressure just by changing vascular size. 2, 4, and 5 increase blood pressure by increasing blood volume and cardiac output. Okay? So increases blood pressure by blood vessel constriction, increases blood volume. Okay? Remember I said this is all downstream? So this is not going to have an immediate effect right there at the filtration membrane, but it has a systemic effect on blood volume and therefore increases capsular hydrostatic pressure. So on Tuesday, we'll look more directly at the effect of aldosterone and ADH, although we've already talked about it briefly. Remember the Mary and her little lamb? So aldosterone increases sodium reabsorption and water follows, therefore there's an increase in blood volume. Therefore there's an increase in venous return. Therefore there's an increase in cardiac output. Therefore there's an increase in blood pressure. Okay? ADH does the same thing but without having to increase sodium. It increases the amount of water that can come across the collecting ducts, and we'll talk a little bit about how that is occurring afterwards. All right, so in the last hour and a half, we have just gotten through filtration. So would you like some study time when you get back and take the quiz then before we move on? Okay. So on your quiz, by the way, as a heads up, there's no histology, but I do ask you to identify these cells. Okay, so from the model, I want you to be able to identify the macula densa. I want you to be able to identify the granular cell and the afferent arterial. I want you to be able to identify podocytes. And if they're seeing the intraglomerular mesangial cells. So on the model here, we would have the macula densa. Okay, macula densa, podocytes, the granular cells or juxtaglomerular cells. Okay, what pole is this? Vascular pole and urinary pole. Okay, even though it's not urine going through there, it's filtrating. All right. The rest of the questions will be from this model or here, asking about the ureter, the pelvis, the minor calyx, the major calyx, the vessels. Okay, and then from that long, colorful one, same thing because you can see it better. So afferent, efferent arterioles, cortical radiate vessels, peritubular capillaries, vasa recta, and the parts of the nephron. So proximal convoluted tubule, capsule, descending limb, ascending limb of the nephron loop, collecting duct, and